and nice to meet you, everybody. Hopefully you can all see my slides. So my name is Jeremy Barr, and I'm currently a senior lecturer at Monash University in the School of Biological Sciences. And I'm going to tell you today a short story. This has been some work that we've been working on over the last couple of years now, looking at the discovery of temperate bacteriophages. And so I want to start really simply with what is a bacteriophage. And put really simply, a phage is a bacterial virus, and it's a specialized virus that can only infect and kill bacteria. And so being a virus is completely dependent on its bacterial host for its own growth and replication. These viruses typically are very specific with strain level specificity. And they're also the most abundant and diverse organisms, not only on the planet, but also in your own bodies. But I want to take it a little bit further. And what is a bacteriophage really, or how do we study them? And these phages come in many different forms, or we can look at them with many different techniques. One of the simplest ways we look at a phage is this viral plaque assay, where a single plaque in this plate will be the result of a single phage propagating, lysing, and killing its bacterial host, and we can count these phages this way. We can also look at how the phage predates and kills its bacterial host in real time through a growth curve. But we can also look at them using microscopy. So we can fluorescently label the phage's nucleic acid, and we can use fluorescence microscopy, or we can use electron microscopy to get these beautiful images of the phage virion structure itself. Of course, we can also extract their nucleic acids. We can extract these, we can sequence them, and we can begin to assemble, annotate, and understand the genome architecture or features of these viruses. And with this information, we can begin to look at the phylogeny, diversity, and how abundant these viruses are in different environments. And we can also do a range of different induction gene expression studies with this data. So we can look at phages in a really diverse mechanism of ways. But studying phages in the gut is actually really, really hard. And one of the issues is that we're largely limited to sequencing and informatic approaches to characterize and understand these viruses. And this is due to one of the limitations that the majority of human gut bacteria don't have any represented cultured isolates. And as such, it makes wet lab characterization, studying these phages in context with their bacterial host, really difficult. However, a lot of that changed over the last couple of years, and in particular, some work by Sam Foster, who's one of our collaborators on this project. Really, what Sam in his lab did was develop a new method or a technique that allows us to culture these sort of unculturable gut microbes. And so Sam's group runs the Australian Microbiome Culture Collection, or AMCC, which has well over 4,000 gut bacterial isolates. This is a phylogenetic tree taken from one of their previous papers, showing the diversity of these gut isolates across the background of the human gut. And so with this collection, we've got three important aspects to consider. The first is that we have access to these cultures, that we can grow them under pure culture conditions in anaerobic incubators, and we have whole genome sequence information. So with this, Sam and I started a collaboration to look at some of the phages infecting these gut bacterial isolates. So we felt 4,000 isolates was a bit too many to work with, so we bit of a smaller number and started working with 252 gut bacterial isolates. And again, we have pure culture representatives, and we can grow these under anaerobic conditions. But we started with their whole genome sequence information, and we took this whole genome sequence information and we used a range of publicly available prophage identification tools, ViaSorter, Vibrant, Finder, to predict and identify prophages or phage genomes that have integrated into the bacterial host. And so I want to take a little bit of a segue and just go through the two major phage life cycles. So the lytic cycle is where the phage injects its nucleic acid into the host, it replicates, produces more phages, and lyses or kills the host. But the lysogenic cycle, here the phage will infect the host, and instead of replicating, it will integrate its genome inside the bacterial host where it resides as a prophage. And this prophage is stably maintained by the bacterium. When the bacterium divides, it also replicates the prophage until the point that that prophage is induced, and it leaves the lysogenic state and enters back into the lytic life cycle. And importantly, we are investigating, or we're characterizing these prophage sequences, these integrated phage genomic sequences that we're predicting or identifying via these informatic sources. And so I've got to give a big shout out to Sophia Dahlman, who's a recent PhD graduate from our lab. This has been her research project. She's generated and led all of this research. And here we've got a phylogenetic tree of our 252 bacterial isolates. These were broadly representative of the major phyla found in the human gut. And you can see these down the bottom here. And taking these genome information and predicting the phages, Sophia found over 1,000 predicted prophages. And these are all shown in these dotted gray circles around this phylogenetic tree. And we found that roughly 95% of gut bacteria harbored one or more predicted prophage, which is quite in line and common with what we know about the human gut virome. However, we wanted to take this a step further. We had these isolates, we had the sequences, we had the predicted prophages. Let's go back to the lab and let's see if we can actively induce these and grow these phages. So we took these isolates, we grow them under pure culture conditions in a specialist media under anaerobic conditions, and then we expose these isolates to a range of different stresses or induction agents. And these stresses are to stress the bacterial host and induce the phage out of that temperate life cycle and back into the lytic cycle to produce these productive or infectious viruses. We then cleaned up the samples. We removed the bacterial host DNA and residual fragments. We then lysed the phage genomes. We did DNA extraction and purification, and then these were sent off for sequencing. And so we took this sequence data and we applied a read coverage and mapping approach to identify active prophages in the sample. And the program that we used for this was Phage. It was developed by these amazing people and collaborators pictured here. And basically what Phage does is it takes this read map or this sequence data and it maps it back to our bacterial host chromosome. And by aligning this read coverage, we can find or we can observe these regions with high sequence coverage that are our predicted prophages. And Phage will use a range of statistical cutoff tools to predict these. Now, obviously, this is sort of hypothetical data. So if we look at one of our genomes, we can see what this actually looks like. And you can see this genome is quite fragmented. And piggyback off Phil's talk, we can see a lot of problems with these fragmented genomes. But even with this fragmented genome, we have our read mapping in the dark black line, and we can see that there's very little reads mapping to the majority of this chromosome. And over on the right here, in one of our final contigs, we have very high read mapping. We've got a clear start and end point, which is this prophage excising, over 50,000 fold coverage. And this was very clearly identified as one of our active induced prophages. So with this approach, if we go back to our predictions, we had 252 human gut bacterial isolates, and we predicted over 1,000 prophages using informatic sources. And if we compare and contrast this to what we actually found, we observed that just 20% of those predicted prophages were active in our sample set under these anaerobic wet lab conditions. We also found that only about 40% of the bacteria harbored an actively replicating prophage, or a phage that could leave that lysogenic cycle and re-enter into the lytic cycle. If we pull apart this phylogenetic tree a bit more, we've got genus and species information on the left. We've got our major phyla shown in the colored branches of the tree here. If we look at the outer ring in this tree, these pink bars are giving us a qualitative assessment of the number of predicted or informatically identified prophages, whereas the black dots are showing the number of active prophages. So just as an example, this isolate here over in the firmicutes had quite a large number of predicted prophages, and indeed we found 4 active phages in the sample set. However, this clustered on the bottom here, you can see quite a large number of predicted, but very few, if any, active phages. Now, we can also see these green or teal lines, and these are showing active phages that are shared across species, and the black lines are showing active phages that are shared across genera. So we have quite a number of broad host range phages that were actively replicating and capable of lysogenizing and spreading across this sample set with these gut bacterial isolates. We can also take all of our active prophages and we can map these on current publicly available gut metagenome and virome samples. So here we've got in gray all of these publicly available genomes and sequences, and in the blue, or sorry, the colored squares are all of our active phages that we found in our sample set. So you can see that we've got quite a number that represent across the diversity of gut viruses known to date. And we identified a number of very high abundance phages. One of the most abundant phages in the body and potentially on the planet is phage. This is found in over half of the world's population in their gut. We found this at a frequency of about 20% in publicly available metagenomes. And below this, we've got all of our colored circles, and these represent a number of phages, particularly in the Bacterioides grouping, that were very high abundance, between 5 and 20%. So these are high abundant phages, and I really want to emphasize these are some of the first cultured representatives. We've got a lot more data characterizing these phages, but I'm completely out of time, so I'm going to wrap up there, and I hope that sort of sparked your interest in the diversity of these gut viruses. I'll give a big thanks to my lab, particularly to Sophia, to Laura and Dinesh, who led a lot of this work, also to Sam, one of our collaborators, and our funders for keeping us running. And thank you all for listening. I hope you enjoyed the talk. Cheers.